Hello again. Now we're going to finish the end of chapter 12. By and large, we have finished chapter 12. This lecture is more to refine our understanding of some of the events that occur in meiosis and also to further differentiate and refine our understanding of the difference of meiosis from mitosis. So here we have a fantastic slide which compares meiosis and mitosis side by side. And the initial thing that you'll notice, of course, is that meiosis has its two divisions, meiosis 1 and meiosis 2, whereas mitosis undergoes only the one division. And you can see at the bottom of the two slides, the divisions of mitosis and meiosis 2 are vastly much more similar. Meiosis is cell division that occurs only during gamete formation. That's it. During meiosis, the genetic material must be reduced by 50% to a haploid state. Meiosis 1, the first cell division, is a reductive division because the chromosome number is reduced to a haploid number in the resulting daughter cells. In meiosis 1, homologous chromosomes, which are made up of two sister chromatids, line up along the equatorial plate during metaphase 1, and then they will separate one homologue from the other during anaphase 1. Meiosis 2 directly follows without undergoing any further DNA replication. During anaphase of meiosis 2, the centromeres are duplicated and sister chromatids will segregate to opposite poles of the cell. Each gamete formed will then contain a haploid genome consisting of 23 chromosomes, and each of those chromosomes then at that point is a single chromatid. There are important events highlighted in meiosis on this slide in orange boxes, and that's going to be where you see the pairing of homologous chromosomes. And those chromosomes, when they pair up in a line next to each other, they form what's called the synaptonemal complex. And this is where synapsis and then crossing over will occur. And we'll go through that much more in depth in a couple of slides. Okay, to review, on the other side, on the right-hand side of this slide, is mitosis. And so before we enter mitosis, in the S phase of interphase, you undergo DNA replication. And then the cell will go through the stages of mitosis, which are the PMAT, the prophase, metaphase, anaphase, and telophase, which are very much like meiosis 2. So during prophase, we're going to have the chromosomes condense up, they'll line up on the metaphase plate, and at that point, sister chromatids are going to separate from each other during anaphase. And in telophase and cytokinesis, we're going to have the cleavage furrow come around, and we're going to form two new cells that have the full complement of DNA. So to re-examine some of the differences here, in meiosis 1, we have homologous chromosomes pairing up, whereas in mitosis, the homologous chromosomes do not ever pair up. In meiosis 1, we have crossing over events where the sister chromatids will swap some DNA, and this does not happen during mitosis. We have two cell divisions in meiosis, both meiosis 1 and meiosis 2, and there's only the one cell division in mitosis. Meiosis will result in the formation of 4 daughter cells, each of which have a haploid nucleus. Whereas in mitosis, we will end up with 2 daughter cells that are exact clones of the parental cells, so each daughter cell ends up with a diploid nucleus. All right, let's now move on to looking at meiosis and those special events like crossing over in more depth. All right, here is an up-close view of meiosis 1. You'll see here during meiosis 1 that prophase 1 is sort of the signature event of the meiotic process because it's here that the genetic recombination events take place. Prophase 1 is really complicated, and in higher level textbooks, you'll even see it is subdivided into 5 phases, but I'm not going to go that in depth for you here. I hear the collective sigh of relief. At the beginning of prophase 1, we can see that each chromosome has a single centromere and 2 sister chromatids. And eventually, the chromosomes come to a point where they associate with each other and pair. And this is where they form that synaptic complex. Okay, it happens along the entire length of the chromosomes, and they coil up and they condense some more. And this synapsis is a really important point of prophase 1 that you need to know. Synapsed paired homologous chromosomes are termed bivalents, indicating 2 joined or synapsed chromosomes, or tetrads, representing the 4 separate chromatids in the bivalent structure. Here on this slide, the term tetrad is used. These tetrads will undergo an exchange of chromatid material in a process of recombination called crossing over. Crossing over is a major source of genetic variation, and it permits an extremely high degree of variability among gametes produced by an individual. After crossing over, when the homologs are ready to separate, we enter metaphase 1, and a spindle apparatus will form and the paired chromosomes align along the equatorial pole of the cell. During anaphase 1, the individual bivalents completely separate from each other. And we see this in these pictures, right? So you see metaphase 1, where they're lined up on the metaphase plate, and we have the microtubules attaching to the kinetochore. And then in anaphase 1, you can see where the sister chromatids remain attached, but the homologous chromosomes separate. So this is the big difference from mitosis. In mitosis, we're actually separating the sister chromatids from each other in anaphase, but in anaphase 1 of mitosis, we're separating the bivalents or the homologous chromosomes from each other. So as you recall, when we did our earlier graphics about homologous chromosomes, we did pink or red for the chromosomes we inherited from mom, and blue for the chromosomes we inherited from dad. And you can see that here in the same picture. So you see the homologs in one, we have it starting to form there at anaphase, where a red chromatid, sister chromatids, are going to one side, and the blue are going to the opposite pole. And then we also see for the larger chromosome, we have the blue one going to the pole, the north pole there, and the red larger chromosome going to the south pole, for lack of a better term on saying this on slides. So what we end up with by the time we end up in telophase and cytokinesis here at the end of meiosis 1 is we have our northern pole cell where the cleavage furrow is forming, where we have our smaller chromosome that appears to have been largely inherited from mom, though it has a little piece of blue on it that had recombined during crossing over. And the larger chromosome is blue, so it was originally a paternal chromosome inherited from dad. And we see a larger section of red on that chromosome on both ends of the chromosome where crossing over events occurred. Conversely, in the south pole cell, we see that the smaller chromosome was inherited from dad and is blue, with that small piece of red from a crossing over event from mom, the opposite of what happened above. And we see the larger chromosome was inherited from mom and is mostly red, except for 2 large ends of the one chromatid that had crossing over events or exchange with a paternal chromosome. Okay, so you can see how there's this random segregation of the chromosomes. We could have had both the large and the small chromosome of maternal origin, or largely red, ending up in the top cell, the north cell, or we could have had, and conversely would have had, both blue chromosomes ending up in the bottom. And this is random. This is the independent assortment of the non-homologous chromosomes. So those are two different chromosomes, both of which could have come from dad, both of which could have come from mom, or as in the cases we see here, one came from mom and one came from dad in each of the cells. And we have further genetic recombination because of the crossing over events that rearranged on those individual chromatids as well. Okay, so let's review our processes once again. The right-hand side of the slide again shows mitosis, which is asexual cell division, starting with a diploid parent cell and ending with exact copies of the parental cell, which are also diploid. Mitosis began with prophase, in which the chromosomes condensed and the spindle began to form as the two pair of centrioles moved to opposite poles and microtubules begin to polymerize from the duplicated centrosomes. We then entered metaphase of mitosis, where the chromosomes assume their most compacted state. During metaphase, the centromeres of all the cell's chromosomes line up at the equator of the spindle. After going through a metaphase checkpoint to make sure everything is correctly assembled, the cells progress from metaphase into anaphase, which is marked by the abrupt separation of the sister chromatids. Mitosis ended with telophase, or the stage at which the chromosomes reach the poles. The nuclear membrane then reforms and the chromosomes decondensed into their interphase conformations. Telophase was followed by cytokinesis, or the division of the cytoplasm into two daughter cells. The daughter cells that resulted from the process of mitosis have identical genetic compositions to each other and to the parent cell. Meiosis, which we just went through in depth, accounts for some of the big occurrences that we will talk further about in genetics: segregation, independent assortment, and the recombination of linked genes on a chromosome. This recombination, or crossing over, was the exchange of genetic material between homologous non-sister chromatids, a process that greatly adds to the genetic diversity by generating new combinations of genes. In meiosis 1, we had the pairing of homologous chromosomes, in which maternal and paternal homologs of each chromosome are replicated and then undergo exact pairing along their lengths during prophase 1 of meiosis 1. This paired unit was either called a bivalent or a tetrad because there are only two centromeres, although it's composed of 4 chromatids. At this 4 chromatid stage was when crossing over occurred. During crossing over, the chromatids from non-sister chromatids, say one chromatid from a paternal homolog, will swap DNA with the chromatid from a maternal homolog. And this swapping of DNA segments causes a lot of genetic variability. Another form of genetic variability comes from the segregation of the maternal and paternal homologs. The centromeres holding the replicated sister chromatids together do not separate at meiosis 1. Instead, the members of a maternally derived homologous pair will separate from the paternally derived homologous pair. When we get into chapter 13, you will see this is how we arrive at and can attribute the cellular events to what Mendel eventually describes as his first law, or the law of segregation. And I'm just planting that first seed for thought in your brain right now. Now, on all these slides, we only see 1 or 2 chromosome pairs. We never show all 23 chromosome pairs in a human. But what you will notice is that at the end of meiosis 1, where we have the segregation of the chromosome homologs, you'll find that paternally derived chromosome homolog will end up in one cell, and the maternal derived chromosome 1 will end up in another cell. And that is independent of the way paternal and maternal chromosome 2 or chromosome 3 will end up in a cell. So a cell can divide where it has all the paternally derived chromosomes, and another cell could end up having all the maternally derived homologs end up in the cell. But what you end up is usually with a mix where, say, you get paternal homolog 1, maternal homolog 2, maternal homolog 3, paternal homolog for chromosome 4, and so forth. And it's random which cell each chromosome homolog ends up being in. And hopefully this will soon start to make sense for you. So this segregation of maternal and paternal homologs is yet another source of genetic recombination. Okay, we then enter meiosis 2, and the second meiotic division occurs without a preceding round of DNA synthesis and chromosome duplication. In meiosis 2, the 2 chromatids of a chromosome will move to opposite daughter cells, and this is how we end up with 1 chromatid in each haploid daughter cell. So again, drawn here at the bottom, you see that meiosis 2 resembles an ordinary mitotic division, except for the presence of a single set of 23 duplicated chromosomes, each with 2 chromatids held together at their centromere. And just to rehash a point, let's look again at keeping track of what is diploid, what is haploid, what are homologs, and what are sister chromatids. So up in our first box, we have sort of the normal state of all the somatic cells in our body. They are diploid, and they each have a chromosome pair or homologs, but our homologs are only made of 1 single chromatid. In the box below that, you have homologs that have been through the process of replication because they're ready to enter meiosis. So they've been duplicated. So we have our 4 chromatids. On the right-hand side, we have a singular haploid chromosome. And that is what appears in a gamete cell. In the box below that, we have a haploid chromosome that has been replicated. And this would basically be what appears in the stage of anaphase 2, before the sister chromatids are pulled apart into separate cells. Okay, so meiosis fosters genetic diversity. Crossovers and that random sorting of chromosomes into the gametes results in new combinations of traits among offspring. And here is a little picture just to remind you of that. You see how we have the bivalents at the top swapping the tips of their chromosomes for crossing over. And so we have totally newly formed chromosomes that are part blue and part red as a result of crossing over. Meiosis also gives rise to variation in traits among offspring by randomly segregating homologous chromosomes into gametes. Microtubules can attach the maternal or the paternal chromosome of each pair to one or the other spindle pole. Each chromosome can end up in any new nucleus and in any gamete. Along with fertilization, which is the fusion of 2 haploid gametes that restores the diploid number, These events will contribute to variation among the offspring of sexually reproducing species. Let's look a little bit at crossing over and how it occurs up close. During the bivalent or tetrad stage, homologous chromosomes are held together close enough such that they are able to physically exchange segments of their DNA. There's a very tight parallel orientation between these homologous chromosomes, and so the partners can exchange corresponding pieces of DNA. They're pretty much, breaks off the end of one and the end of another one, and they can kind of overlap and splice together. And we find that crossover events happen more frequently in certain regions of DNA. You can see at the end of crossing over, the resultant chromosomes are neither entirely maternal nor entirely paternal, but contain genes from both parents. This slide is a review of how we can account for genetic variation. We have the independent assortment of chromosomes, crossing over, and then random fertilization. And you can look at this slide and review each phenomenon. We'll look at each one more in depth in the following slides. Again, crossing over is the process by which a chromosome and its homologous partner exchange segments of DNA, and this always occurs in prophase 1. Looking at it up close, you can see we have the blue chromosome represented with genes containing all capital letters, and the pink chromosome from the mother is represented by genes containing alleles that are all represented by small letters. During a crossing over event, the gene contained from the pink chromosome is recombined onto a blue chromosome, and similarly, a little tip from the blue chromosome is recombined onto the pink chromosome. And this happened in an area containing the B gene. And we see with the new recombined chromosome or chromatid in the third panel, you have one that remains a completely parental chromatid, having both capital A and capital B. And then you have a completely new chromatid, which contains the paternal capital A gene or allele, but it now has the small b maternal allele on it. Similarly, on the pink chromosome, you have one chromatid in which both have the maternally derived small letters representing different alleles for a gene. But on the other chromatid, you have one maternally derived small a allele and a paternally derived capital B allele. Now let's look at chromosome segregation and how this fosters genetic diversity. Homologous chromosomes can be attached to either spindle pole in prophase 1, so each homolog can be packaged into either one of two nuclei. By metaphase 1, the homologous chromosomes have been divided up between the spindle poles in one of four ways. In anaphase 1, maternal and paternal chromosomes separate and move toward opposite spindle poles. And in telophase 1, a new nucleus forms around each cluster of chromosomes. Each contains one of 8 possible combinations of maternal and paternal chromosomes. Let's look at this in a picture. A picture is always better for these things. Here you see the homologous chromosomes of 3 chromosome pairs that can be divided between 2 spindle poles in 4 possible ways. In the first panel, you see all blue to the left and all pink to the right, or all paternal derived chromosomes to the left and all maternal chromosomes to the right. In the second panel, you see the first chromosome is a maternally derived chromosome going to the left, and the other chromosomes are paternally derived. Conversely, going into the right side of the cell, you have one paternally derived small chromosome, and the two larger chromosomes are maternally derived, and so forth. I don't think I need to go at it ad nauseum. And you will see that by the time we're splitting apart the sister chromatids as we get down to telophase 2, we will have 8 possible combinations of maternal and paternal chromosomes that can end up in the 4 different nuclei that are forming at telophase 2. Each nucleus divides again at meiosis 2. Human germ cells have 23 chromosomes, so each time one undergoes meiosis, each of the 4 gametes that form have one of 8,388,608, or 2 raised to the 23 possible combinations of homologous chromosomes. A huge number for genetic diversity. This is why none of us end up being the same. In addition, any number of genes may occur at different alleles on the maternal and paternal chromosomes, and crossing over makes mosaics of that genetic information. So keep track of the ploidy, or the number or sets of chromosomes in a cell. Is it haploid or N, or is it diploid or 2N? Versus the number of chromatids, or the DNA content, which is C, okay? Meiosis 1 reduces the ploidy level from 2N to N, or from diploid to haploid. It is the reductional division. Meiosis 2 is akin to mitosis, and this involves an equational segregation or separation of sister chromatids. So again, diploid, 2N, and here we have 2 chromatids for that. In the bottom panel, this is where we had replicated our DNA. So we have 2 sister chromatids forming each chromosome. We have the same number of chromosomes, 2N or diploid, but we have double the DNA content, or double the number of chromatids. And again, here is another picture that might help you, comparing meiosis 1 and mitosis. Both processes are illustrated with a diploid genome composed of 2 homologous chromosomes, one maternal one represented in red and one paternal one represented in blue. And you can see in meiosis 1, we have the pairing of homologous chromosomes that are then separated. Okay? They're separated at the end of meiosis 1. Then meiosis 2 will take those cells and do an equational division, which splits the sister chromatids apart. Thus, at the end of meiosis 2, we end up with 4 haploid cells that are 1N and also 1C, or 1 chromatid. In mitosis, we start out with a diploid cell and we replicate our DNA content. And then we undergo our division where everything lines up on the metaphase plate, and then we move to split apart our replicated or sister chromatids in mitosis. In anaphase, we have the separation of the 2 chromatids to the separate poles, and we then form 2 new cells, daughter cells, which are diploid and exact replicas of the parental cell. So as I said, in mitosis, the chromatids of both chromosomes are separated and distributed to the 2 daughter cells. In the subsequent interphase, the DNA is replicated, completing the complementary chromatid. And that is what you see here under the arrow with the S phase, where the chromatids were replicated. You then have a diploid cell, which has a 4 times the DNA complement, or 4 chromatids at that point. It then goes through G2 and enters then the mitosis process of prophase, metaphase, anaphase, telophase in cytokinesis, where we're going to have the chromatids separating back into the two daughter cells and restoring the diploid 2C state. In meiosis, we have two sequential cell divisions that result in 4 daughter cells. In meiosis 1, the homologous maternal red and paternal blue chromosomes are paired, then they are separated and distributed to the 2 daughter cells, which are now haploid but still have double the DNA content because you have 2 replicated chromatids. This is the point at which crossing over can occur during prophase and metaphase, most likely during prophase 1. Meiosis 2 will then proceed like a mitosis in which the chromatids are then distributed to the daughter cells. So now we end up with 4 cells that are haploid and have 1 chromatid in them. Let's look at this again in a picture. And here you go. We start out with our diploid germ cell, which is 2N and 2C. We undergo the S phase where we're replicating our chromatids, so we end up still being diploid, but we have a 4C DNA complement. We enter prophase, where synapsis and crossing over can occur. And then at metaphase, the tetrads, or the bivalents, will line up on the metaphase plate. The separate homologs, the parental homologs, maternal or paternally derived, will then go to their separate poles at anaphase. And they are completely separated at telophase, and we have cytokinesis 1. At this stage, we end up with cells that are haploid, but they still have twice the DNA content because we still have those replicated chromatids in there. This is where we're at when we're entering meiosis 2, which is the prophase 2 stage. Then meiosis 2 continues much like mitosis, and we have our chromosomes again enter metaphase 2 and line up on the metaphase plate. And now we're going to pull apart our sister chromatids. And the sister chromatids go to opposite poles at anaphase 2, and then at telophase 2, we start forming our 4 individual cells that are now haploid and only have 1 chromatid representing a chromosome in each cell. The 4 cells will then undergo cytokinesis 2, resulting in the 4 haploid daughter cells that are formed. And I've told you previously that in spermatogenesis, we form 4 complete daughter cells, 4 individual spermatids. When we're forming the female gamete, the ovum, only one of these ovum will get all of the cytoplasm from cytokinesis. The 3 other cells will really get none of the cytoplasm, and they'll degenerate as being what's called polar bodies. Though they have different results, mitosis and meiosis are fundamentally similar processes. The earliest eukaryotes were single-celled and haploid, and they reproduced by mitosis. Meiosis evolved later from mitosis. As your textbook shows, mitosis and meiosis use the same molecular machinery. Hence, there is an ancestral connection. Though the end results differ, there are striking parallels. The 4 stages of mitosis and meiosis 2 are quite similar. Long ago, the molecular machinery of mitosis became remodeled into meiosis, and evidence for this hypothesis includes a host of shared molecules, including the products of the BRCA, the BRCA genes, that all modern eukaryotes have. By monitoring and fixing problems with DNA, such as damaged or mismatched bases, these molecules actively maintain the integrity of a cell's chromosomes, particularly during DNA replication and mitosis. Many of the same molecules also help homologous chromosomes cross over in prophase 1 of meiosis. This is another example of shared molecules, the same regulatory molecules being involved in checkpoints of mitosis and also in meiosis. The same mechanisms that operate during meiosis also operate during mitosis, and many of the same molecules function the same way in both processes. These similarities are evidence that meiosis evolved from mitosis by repurposing an existing molecular system. And here is the picture in your book, which is a fluorescent view of the location of a protein called MLH1. And MLH1 has several functions in meiosis, including chromosomal repair after crossovers and preventing inappropriate crossovers. But this protein also has several functions in mitosis. For example, it helps repair DNA replication errors, and it prevents homologous chromosomes from crossing over. So, very interesting. All right, let's go through a few practice questions, and then we can end this chapter. In which of the following processes do the chromosome and its homologous partner exchange corresponding pieces of DNA? Crossing over, gametes, alleles, haploid, or germ cell? There's only one obvious answer, and that is crossing over. The production of which cell type requires a different nuclear division mechanism that halves the chromosome number? Somatic cells, gamete cells, prokaryotic cells, zygote cells, or stem cells? And of course, the answer is gamete cells. Gamete cells require meiosis 1, which has the reductional division. If meiosis did not precede fertilization, what would happen to the chromosome number after each generation? Would it be half the parent's chromosome number, or twice the parent's chromosome number, or 4 times the parent's chromosome number, or 8 times the parent's chromosome number, or would it simply be the same? And it would be twice the parent's chromosome number, because we never underwent a reductional division. So we'd always be increasing or doubling. And so you could see how that would get out of control. Meiosis is cell division that occurs only during gamete formation. And we just did a question like this, and it is true. Meiosis is cell division that occurs only during the formation of gametes. Crossing over. This is basically a question as to what is the purpose of crossing over. Does it happen only once per homologous pair? Is it followed immediately by separation of chromatids? Does it increase variability in gametes, prevent genetic recombination, or happen only once per homologous pair? I guess I duplicated on that one, sorry. And it obviously increases variability in gametes. It can happen more than once per homologous pair. It is not followed by separation of the chromatids. It's followed by separation of the homologs going to opposite poles. And it does not prevent genetic recombination. It encourages it. Crossing over mixes up chromosomes, alleles, zygotes, or gametes. And crossing over will reshuffle all the alleles. It's going to mix up the genes, the alleles of the different genes. It doesn't actually mix up or change over chromosomes. You still have 23 chromosome pairs, right? Or 23 chromosome haploid chromosomes in a gamete. Okay? Which phase of meiosis reduces the chromosome number? So this is the reductional division. Okay? And the reduction happens during anaphase 1. Anaphase 1 is when they're actually separated at different poles. Genetic recombination can result from either independent assortment, 50% recombination frequency, or crossing over, recombination frequency somewhere between 0 and 50%. True or false? And this is true. Our genetic recombination results from the independent assortment of the chromosomes. We saw that, 2 raised to the 23, or by the recombination occurring through crossing over events. During meiosis, independent assortment occurs in which stage? Meiosis 1, meiosis 2, mitosis, or cytokinesis. And all those recombination events are really occurring during meiosis 1. It's meiosis 2 that is the equational division, which is a lot more like mitosis. And we don't have those recombination events occurring either in mitosis or in meiosis 2. The number of chromosomes does not correlate with the apparent complexity of an animal or a plant. In humans, for example, the diploid number is 2n equals 46, or 23 chromosome pairs, compared with 2n equals 78 in a dog. How many chromosomes does a dog gamete carry? Well, we have the example right here, so you should be able to figure out that the haploid number for a dog would be 39. Okay, that's the end of this chapter. I hope you study well and do your mitosis and meiosis module questions and get them into me. And then we'll move on to chapter 30.